Hi friends, welcome all of you to the class of Dental Plaque. I hope you all are doing great. For those who are interested in these type of classes related to dentistry, please subscribe to my channel and turn on the bell icon for notification. And it would be great if you like the video, which is definitely an inspiration for me to proceed further. Let's begin. Dental plaque. First, we will understand what is the definition of dental plaque. Dental plaque is the structured, resilient, yellowish gray substance that adhere tenaciously to the intraoral hard surfaces. Within hours after the birth, we can see the aerobic bacteria and it uses all the oxygen, and by the second day we can see the anaerobic bacteria, and after 2 weeks mature microbiota can be seen in the gut, and usually the bacteria are 10 times more than cells. So these are about the bacteria in our body. Then we will see the classification of soft deposits. So there are four types of soft deposits are there. First one is the acquired pellicle, then the food debris, material alba, and dental plaque. So we will see what all are the differences. There will be only slight differences between these four, so that it would be little difficult to understand. So we will see what is acquired pellicle. These acquired pellicle, they come immediately after we brush our teeth. So they are the specific carbs or specific proteins. They come from the process of adsorption. So this may take approximately 30 minutes to make the acquired pellicle. This is the layer which stick on the surface of the teeth. So something which helps the bacteria to hold on the tooth. So this is the acquired pellicle. Then the food debris. If you know, food debris is just a remnant of food. Usually saliva just flush out the food debris. Then material alba. This is the white soft deposit accumulate on the tooth. If you don't brush more than 12 hours, then this material alba can be seen on the surface of the teeth. This may not be removed by the help of saliva. This usually removed by water spray only. Needs some pressure to go, not just with the saliva. And this material alba lack the organized structure of plaque. It's just a white cheese-like accumulation. So this is the material alba. Then is the dental plaque. Dental plaque is usually after 24 hours of, if you don't brush after 24 hours, this dental plaque start to accumulate. They are the organized deposit which consist of bacteria and the products of bacteria. This is impossible to remove by rinsing or sprays. Then we will understand what are the classifications of dental plaque. So there are usually three types of classification. First type is the coronal, gingival, subgingival, fissure, peri-implant. These all are the types. Coronal means which can be seen on the surface of the teeth. Gingival means on the gingival sulcus. Subgingival means this plaque can be seen inside the gingiva. Fissure type is that the dental plaque on the occlusal surface of the teeth. Peri-implant means plaque around the implant. Then the other type is the supragingival and subgingival. And the supragingival also, the plaque can be seen above the gingiva. Subgingival means inside the gingiva. And above the gingiva, again it is divided into coronal, marginal and fissural. And subgingival is divided into tooth associated, tissue associated and unattached plaque. So tooth associated means, see, if this is the tooth and this is the gingiva, and this area is there, no, above the tooth. This is the area where the tooth associated plaque occurs. So usually tooth associated plaque, the bacteria will be the gram positive bacteria. Then tissue associated means that will be the plaque would be inside the gingiva, so that the bacteria usually present are the gram negative. and unattached can be seen here. So here also the bacteria would be gram negative. Then the other type of classification is again supragingival and subgingival, but supragingival is divided into gingival 1/3, interproximal and pit and fissures, but subgingival is divided into tooth attachment zone, epithelium attachment zone, non-adherent zone. The concept is the same, but the term are little different. That's the change that can be seen in the in this type of classification. And when we see the this picture, you will understand this is the tooth surface, and there's a junctional epithelium, gingival sulcus, sulcular epithelium, outer epithelium. These all are the can be seen in this picture. So when we discuss about the tooth associated plaque, this is the plaque area where the plaque accumulate on the surface of the tooth. So these tooth associated plaque, they doesn't extend to the junctional epithelium. It just stop on the surface of the tooth and forms calculus, root caries, and it may penetrate to cementum. So I just discuss now that the bacteria on the tooth associated plaque are the gram positive. Then next is the unattached plaque. This is the unattached plaque. So this is neither associated with tooth nor associated with the epithelial lining. This is seen in between. So they are usually gram negative. This can extend to junctional epithelium. So this plaque is in the gingival sulcus. This can lead to gingivitis. Then epithelium attached or tissue attached plaque. So this goes inside the epithelial tissues. This also gram negative filamentous bacteria, and this also can extend to junctional epithelium, and that can causes the periodontitis and gingivitis. It can go to connective tissue also. Then next we will see the composition of plaque, usually divided into solid and water. About 70 to 80% are water, 20 to 30% are solid, and in the solid again divided into microorganism and intercellular matrix. Microorganism we can see bacteria and non-bacteria. In matrix we can see organic and inorganic. Organic components are carbs, proteins and fats. Next is the development of plaque. Usually starts with the first stage is the formation of pellicle on the tooth surface. That is the initial stage. After that, initial adhesion and attachment of bacteria. Then the last stage is the colonization and plaque maturation. So first we will see how the pellicle formation occur. This pellicle, I told you before about the acquired pellicle. This is not comprised of any cells. It is just the connection between the tooth and the plaque. They are only the selective proteins or carbs bind to the surface. The significance of this pellicle is that this is just the moist layer which prevent drying. So this is actually the pellicle is the from the saliva or GCF or bacteria. Some protuberances or attachment occur on the surface of the teeth so that the bacteria can bind to the surface. So that is the pellicle. Then next is the initial adhesion or attachment of the bacteria, because bacteria has to reach to the surface by transportation. So bacteria was just flowing in the saliva actually, and they come and contact to the saliva through sedimentation and Brownian motion, and this bacteria accumulation that can so when they see this bacteria, they see the pellicle. When the bacteria see the pellicle, that is protuberances, they come and attach there. So this is the transportation. Then how it attach by means of the so this bacteria, no, they attach to the pellicle, just close to the pellicle, and they make an attachment. So each bacteria has the specific receptors on the pellicle. So receptors of pellicle we can call as SAP, like sialic acid, amylase, and proline. These are the receptors, but these are for only for specific bacteria, like AS. We can see, like remember, like AS, like actinomyces and streptococcus group of bacteria. So these bacteria, they has fibrils, so they bind to the proline rich receptors. So this is how the attachment occurs. So each bacteria has the specific receptors and they attach with the help of fibrins. So we can say these two as the primary colonizer. So if this is the pellicle and these bacteria like actinomyces and streptococcus, they attach to the pellicle. So we can say they are the primary colonizer. Then next is the colonization and plaque maturation by the help of two mechanisms like co-aggregation and co-adhesion. So I just discussed now the primary colonizer are the actinomyces and streptococcus. So this is the pellicle, and primary colonizer are these two. They are aerobic. They all use all oxygen. Then the secondary colonizers are the Fusobacterium nucleatum and Prevotella, P. intermedia, P. gingivalis. So they are the secondary colonizers. So they don't come and directly attach to the pellicle, but they attach to the primary colonizer. So this is like if this red dots, they are the primary colonizer. Those the secondary colonizer are the yellow ones. So they attach to the red bacteria. So they attach to the primary colonizer. So they cannot attach to the pellicle. So these secondary colonizers are anaerobic because all the oxygen vanished. So they don't require oxygen to grow. So this is the primary colonizer and the secondary colonizer. So this is the subgingival microbial complex. So we can see actinomyces species, streptococcus species. So these all are the early colonizer, and the P and F group, P intermedia and F Fusobacterium group. So these all are the late colonizer, and this is the this is red complex because these causes the bleeding of the gingiva. P gingivalis, T forsythia, and T denticola. So these all, they cause the bleeding on probing. So this is the late colonizers. They are tissue associated and gram negative bacteria. So these all are all about plaque. And there are some questions which have asked for AFK. I have written here. So if you want to discuss this, means I will discuss. Otherwise you can write it down from this. If you want to discuss, means you can comment me so that I can answer these questions. Thank you.